This method enables exploration of specific T and B cells at the nucleotide or amino acid level, and thus can identify dynamic changes in lymphocyte populations and diversity parameters in different disorders.
The advantage of next-generation sequencing is the ability to identify unique T or B cell clones and track them in different anatomical sites or in different individuals. This has the potential for identification of novel biomarkers in diverse diseases.
Demonstrating the procedure will be Naomi Salamon, a research assistant from my laboratory.
Begin by performing digestion and cell lysis of intestinal biopsies. Retrieve intestinal biopsies, either freshly collected or stored at -20 or -80 degrees Celsius. If using frozen biopsies, thaw them on ice. Add 600 microliters of nuclei lysis solution to a sterile 1.7 milliliter microcentrifuge tube chilled on ice. Then, place a biopsy into the tube and incubate it at 65 degrees Celsius for 15 to 30 minutes. Add 17.5 microliters of 20 milligrams per milliliter proteinase K and incubate the tube overnight at 55 degrees Celsius with gentle shaking. After the incubation, allow the sample to cool to room temperature for 5 minutes. For DNA isolation from blood, gently rock the tube until it is thoroughly mixed. Then transfer blood to a sterile 15 milliliter centrifuge tube containing 9 milliliters of cell lysis solution. Invert the tube several times during a 10-minute incubation period